Hello students, very good morning. I am Ashutosh Basti from Tirthankar Mahavir College of Agriculture Sciences, and now we will discuss the process of respiration including glycolysis, TCA cycle and electron transport system. Respiration releases the energy. What we eat or what type of food are stored in our bodies, it has energy. And the process that extracts the energy from this food is the respiration. Because the molecules have a lot of energy, and if that energy comes out of that molecules in an uncontrolled way, it can harm the cell. Therefore, the respiration developed as a very controlled process. Here energy is generated step by step. And the chemical reaction that you can see in my screen, where a carbohydrate in presence of oxygen changes into carbon dioxide and H2O. It includes more than 50 reactions. Mainly the respiration has three main processes: glycolysis, TCA cycle and electron transport system. The glycolysis involves a series of reactions carried out by a group of enzymes. Here, sugar is oxidized into an organic acid, mainly pyruvate, and in this process, small amount of energy in form of ATP and the reducing power NADH is released. In second step, the pyruvate that is generated in the glycolysis enters the mitochondria and it is oxidized into carbon dioxide, and it generates a considerable amount of reducing power, including NADH and FADH2. And we will see in the next step, that is electron transport chain, where this reducing power is used for making the currency of energy, that are ATP. The reactions of glycolysis can be divided into two processes. One is preparatory phase, where energy is not released, but two molecules of ATP are used. And second phase is payoff phase, where energy is released and reducing power is also released. There are 5 reactions in preparatory phase and 5 reactions are in payoff phase. In first step, glucose is phosphorylated into glucose 6 phosphate. Then glucose 6 phosphate is converted into fructose 6 phosphate by enzyme isomerase. In third step, fructose 6 phosphate takes another phosphorus and makes fructose 1,6 bisphosphate. Then in fourth step, fructose 1,6 bisphosphate changes into glyceraldehyde 3 phosphate and dihydroxyacetone phosphate. And in fifth step, dihydroxyacetone phosphate again changes into 3-glyceraldehyde 3-phosphate, and this preparatory phase is now complete. In payoff phase of glycolysis, glyceraldehyde 3-phosphate changes into 1,3-bisphosphoglycerate, and it releases reducing power in form of NADH2. Again, in next step, 1,3-bisphosphoglycerate converted into 3-phosphoglycerate, then 2-phosphoglycerate, and in the next step it is converted into phosphoenol pyruvate. Then in the final step, phosphoenol pyruvate is converted into pyruvate with production of ATP from ADP, and the process of glycolysis is complete. In this way we can see that we have used 2 molecules of ATP in preparatory phase, and we found 4 molecules of ATP in payoff phase. Then the net gain of ATP in this process is 2, and we have one more currency of power in form of NADH that will also be used for making ATP. In the next step, the pyruvate enters into mitochondria from cytosol, and it converted into acetyl coenzyme A. In this process, it releases a carbon, and it also reduces the NAD into NADH. Then this acetyl coenzyme A enters the TCA cycle. TCA cycle is known as tricarboxylic acid cycle because it is conducted by 3 carbon cycles mainly. It has again 10 steps. Acetyl coenzyme A converted into citrate, then isocitrate, then again another molecules, and in this process it releases the reducing power in form of NADH and also makes ATP or GTP, and one more reducing power in form of FADH2 in 6th step. The final step of Krebs cycle is where oxaloacetic acid is made, and again it combines with acetyl coenzyme A and starts new cycle. In this process we can see that two molecules of carbon are released, carbon dioxide, and the final process of respiration is electron transport chain. Electron transport chain of plant contains a set of electron carriers that are enzymes having metals, and these enzymes are found in complexes like complex 1, complex 2, complex 3, complex 4, and one another complex known as ATP synthesis. In this process, the molecules that are made in glycolysis and TCA cycle are again oxidized, and in this process, like photosynthesis, the light reaction of photosynthesis, the protons are exerted out of the membrane, and when they come back through ATP synthetase, here they make ATP. That is the currency of energy per se. The whole process is summarized here. The complex 1 is NADS dehydrogenase. Electron from NADS generated in the mitochondrial matrix during TCA cycle are oxidized by complex 1. 4 protons are pumped from the matrix to the intermembrane space. And complex 2, that is succinate dehydrogenase, it uses FADS2, but in this process no proton is pumped out of the membrane. In the complex 3, it oxidizes ubiquinone and transfers the electrons to cytochrome C, and 4 protons per electron pairs are pumped by this complex. And in the 4th complex, that is cytochrome C oxidase, here oxygen is reduced for making water, and 2 protons are pumped per electron pair. These all protons come back into the lumen through ATP synthetase, and for each ATP synthesized, 3 hydrogen atoms that are protons pass through the F0 from the intermediate space to matrix. And this is the whole process of generating the energy, that is respiration. Thank you.